Hello, grade 11 students. In this video, we'll start with the biology summary for semester 1. Let's start with part 1. In this part, we will start with chapter 1, the biological diversity and the uniqueness of individuals. We'll start with activity 2 about polymorphism. Let's move first for this big question. Where is the genetic information found? This is the big question. We're going to answer this in the coming slides. How did the scientists prove that the nucleus is the carrier of the genetic information? So nucleus carries the genetic information. They have, in this experiment, we have a black mouse, a white one, and a gray one. What did they do? They get the animal cell, a cell from the black mouse, the nucleus of a cell of a black mouse. We get the cell from the white, so we extract the nucleus from the black, so we get only the nucleus from the black mouse, and then we inject it into the cell that is anucleated. We get rid of the nucleus here, and we transfer the nucleus, or we move the nucleus toward the cell of the white, so we injected into the white cell. This white cell, the nucleus was removed, so we inject the nucleus from the black. So the nucleus here is from the black mouse, and then we added this into the cell, or we transplanted into the gray mouse. So the result is bird of the black mouse. So we have a black mouse. So after injecting the nucleus of the cell of a black mouse, so we get the nucleus from the black mouse and injecting it into anucleated cell. What the meaning of anucleated cell? Meaning that a cell that is deprived of the nucleus, without a nucleus. Anucleated cell of a white mouse. So the white mouse, the cell has no nucleus. So we inject the nucleus from the black one, and we transplant the zygote that is formed into the gray mouse. The result was the black baby mouse is born. So we have a black mouse. What does this indicate? This indicates that the carrier of the genetic information is the nucleus, and the genetic information coding for the fur, it doesn't depend on the cytoplasm. Cytoplasm means the cytoplasm of the white mouse, or it doesn't depend on the uterus. The uterus is from the gray one, so we get the uterus from the gray. The gray was the role of the foster mother, so it carries only the zygote. So the genetic information coding for the fur color doesn't depend on the nucleus or the it doesn't depend on the cytoplasm or the uterus. It depends on the nucleus of the cell. So the nucleus of the cell contains the genetic information that codes for the fur. So here, as a conclusion, we deduce that the nucleus is the carrier of the genetic information. So as a summary, this experiment was done to prove that the nucleus is the carrier of the genetic information. So we inject the nucleus of the cell of a black mouse. So we get the nucleus of the cell of the black mouse. We inject it into an anucleated cell of the white mouse. So we get the white mouse, we removed its nucleus, so it becomes anucleated, and then we inject the nucleus from the black mouse into this anucleated cell of the white one, and then we transplant the zygote into the gray mouse. The gray mouse, it has the role of the foster mother. It only gives the uterus. So the color of the mouse that is formed is black. This means that the genetic information that goes for the fur color, it doesn't depend on the cytoplasm from the white. It doesn't depend on the uterus from the gray, but it depends on the uterus of the gray. It doesn't depend. It depends here on the nucleus of the cell that is injected from the black. That's why we have a black mouse. So the result here is the nucleus is the carrier. Chromosomes. What are chromosomes? Chromosomes, they are X-shaped structures. They look like an X shape. So they are called X-shaped structures. They are found inside the nucleus, and they are responsible for carrying the genetic information. The chromosome is composed of short arms, like this. These are the short arms. They are called short arms. They're called P arm, or long arms. They are called Q arms. Here in the middle, the nucleus is connected in the middle with something called centromere. And these two, they are called sister chromatids. This is the structure of the chromosome. It's called X-shaped structure because it looks like an X. That's why it's called X-shaped structure. So this is the structure of chromosome. As I said, these are the short arm. They are called P arm. This is the centromere in the middle. This is a constricted region in the middle. That is the site of attachment of two sister chromatids. This is the long arm. They are called Q arms, and these are the two sister chromatids. They are attached. This is the first sister chromatid, and this is the second sister chromatid. They are attached in the middle, the center, by the centromere. And here's the structure of the chromosome. So, structure of chromosome. Let's move to talk about the structure of a chromosome. Each chromosome is made up of DNA and proteins, histones and non-histones. We're going to talk about this later. Chromosome is made up of two identical, similar sister chromatids. They are joined together at this constricted region, which is called the centromere, which is the site of attachment of two sister chromatids. These two sister chromatids, they are attached in the middle. This is called centromere. Each chromatid has a short arm. As I said, short arm is called the P arm, and long arm is called the Q arm. Centromere, as I said, it's the constricted region near the center of the human chromosome. It's the attachment site where the two sister chromatids, they are attached. We call it centromere. Here, the centromere is now like this. This is a real chromosome. So, centromere, as I said, is the site of attachment of two sister chromatids. Move now to talk about genes. Genes, they are located on the chromosomes. For example, here we have hair color gene, eye color gene, height gene, gene 4, gene 5, gene 6, and so on. So these genes, they are located on two sister chromatids. So they are found at specific locus. Okay, they should be on the same loci. So here we have hair color gene on the first chromatid. It should be the same in the second chromatid, in the same locus, in the same position. Eye color gene is present here, so it should be present in the same locus on the second chromatid. Also for height gene, it's gene number 4. They are present in the same locus. Gene 5, gene 6. Let's move to talk about genes. What are genes? A gene is a part of a chromosome. It's a segment of DNA. It's responsible for the appearance of a specific trait. Like, for example, the hair color gene. It determines the hair color, skin color gene, eye color gene, and so on. Every gene has a specific locus, meaning that a specific position. A specific position of a gene on a specific chromosome. This is called the locus. The two sister chromatids of each chromosome, they carry the same alleles of the same gene at the same loci, at the same position. Here we have the two sister chromatids, they carry the same information of the same loci. So here we have the gene, for example, of the hair color. It should be here of the same color. That's why we can draw it as one, because they carry the same information at the same loci. Let's move now. What are the alleles? Allele is a version, or it's a form of a gene. For example, gene of the eye color, we have the brown eye allele, we have the hazel eye allele, we have the blue eye allele, we have the black eye allele, we have the green, and so on. Gene of the hair color, we have brown allele, blonde allele, red allele, black allele, and so on. So the allele is a version, or it's a form of the gene. Let's move to this table showing the different type of cells and their different properties. Let's start step by step. So in this table we have two types of cells. We have something called somatic cells, and we have something called sex cells, or called reproductive cells. Somatic cells, they are diploid cells, whereas the sex cells, they are haploid. This is the first difference. Second, somatic cells, all the body cells, they are somatic, except for the gametes. The gametes are not somatic. So all the body cells, skin cells, muscle cells, and so on, all of them, they are somatic. Moving to the sex cells, the gametes. Gametes, like the sperm and the ovum, they are example about the sex cells. So the sex cells, they are gametes, the sperm and ovum. Sperm in male and ovum for sure in female. Somatic cells, they have 2 copies of each chromosome, whereas sex cells, they have only 1 copy. That's why the somatic cells, they have 2n number of chromosomes, whereas the sex cells, they have only 1n as number of chromosomes. In humans, somatic 2n equal to 46, whereas sex cells 1n equal to 23. So here the differences between somatic cells and sex cells. Let's repeat them. So somatic cells, they are diploid cells, all the body cells except gametes, whereas the sex cells, they are haploid, the gametes only the sperm and the ovum. In the somatic, we have 2 copies of each chromosome, whereas in the sex cells, we have 1 copy of each chromosome. Number of chromosomes, it's 2n in the somatic, whereas it's only 1n in the sex cells. 1n, it's equal, and 2n, sorry, in somatic, it's equal to 46, whereas in humans, the sex cells 1n equal to 23. Let's move now. In diploid cells or diploid cells, there are 2 copies of each chromosome. There are 2 copies of each chromosome. They carry the same genes, but they don't have the same alleles. They don't necessarily have the same alleles. So they are said to be homologous chromosomes. So in diploid cells, there are 2 copies of each chromosome. So chromosome number 1, we have 2 copies, chromosome number 2, and so on. So they carry the same genes, but they don't have necessarily the same alleles at the same locus. So they are said to be homologous chromosomes. These are homologous chromosomes. One of these chromosomes, since we said that we have 2 copies of each chromosome, one of these chromosomes is inherited from the mother. So one of these is inherited from the mother. That's why M, mother, maternal. So mother, maternal, mama. The other is inherited from the father. Father, it's papa, so it's paternal chromosome. So one of these chromosomes is inherited from the mother. It's called maternal. The other one is inherited from the father. It's called paternal chromosome. So one of these chromosomes, this chromosome, is paternal one, is inherited from the father. The second is maternal, is inherited from the mother. So these are the homologous. So here, when the alleles, they are identical, they are the same. So the individual is said to be homozygous, pure, or homozygote. Or when the alleles, they are different, the individual is said to be heterozygous or hybrid. So heterozygous and hybrid, two different alleles. Whenever we talk about same alleles, homozygous or pure. Let's move to the different types of alleles. In alleles, we have dominant allele and something called recessive allele. Okay, so recessive allele and dominant allele. Let's start step by step. Recessive allele and dominant. Let's start with the dominant. The dominant is the one that dominates. In biology, it's the strong one. The strong one is called the dominant. In English, it's called the strong. In biology, you have to call it the dominant allele. So dominant allele is the allele that will always be expressed whenever in a homozygous, whenever we have two identical alleles, or we have two different allele, heterozygous or hybrid. That's why it's usually symbolized by a capital letter. This is the strong one. So whenever we have dominant and recessive, one dominate over the other. So dominant, it can be expressed either in the case of homozygous or in the case of heterozygous, and usually it's symbolized with a capital letter. Recessive allele. Recessive is the weak one. Recessive allele is the allele that will be expressed in homozygotes only. It should be present with another weak allele that is usually symbolized by a. What is the genotype? Genotype, this is Freyer's model. Freyer's model talking about, here we have always the definition here. Here is always the definition. Here we have the example. And here we have non-examples. This is not an example. Okay? So you have to be familiar with the Freyer's model. This is example, and this is not an example. So genotype is definition, is the set of genes and their corresponding alleles. The genotype, example, BB, BG, GG. Let's move now. Which one of them is pure? Which one of them is hybrid? So BB is a pure, since I have two identical alleles. GG, homozygous or pure, while BG is heterozygous or is called hybrid. This is not an example. Blue and tall. Who can give me the name of this blue and tall? They are genotype? No, they are what? They are the phenotype. They are the observed traits. They are the phenotype. Let's move now to the definition of phenotype. Phenotype, as I said, is the set of observed trait. The set of observed trait resulting from the expression of the genotype. So the genotype will give us the phenotype. Phenotype, tall, short, as I said, this is an example. Tall, short, black, green, whatever. This is non-example. This BB, BG, GG, they are the genotype. Okay, so you have to be familiar with phenotype and genotype. So as a summary, phenotype and genotype. Genotype is the set of genes and their corresponding alleles. This is the genotype. And the phenotype, it's the set of observed traits resulting from the expression of genotype. What is a graft? What is a graft? Graft is always the transplant of a tissue or an organ from one side to another in the same body, or from one body to another. Let's move now to the graft. What affects the fate of the graft? The fate of the graft is affected by the degree of similarity of HLA, human leukocyte antigen, between the donor and the recipient. What does this mean? This means that the fate of the graft, whether it's success or failure, it depends on the similarity of HLA. If you have similar HLA, meaning that the graft is accepted. We have total differences in HLA, they are totally different between the donor and the recipient, so the graft will fail. So the fate of the graft, the result of the graft, is depending on the similarity of HLA between the donor and the recipient. This is the donor, the one that donates or gives. For example, we have grafting of a kidney between Muhammad and Fatima, let's say. So between Muhammad and Fatima, Muhammad is going to give the kidney. So Muhammad is called the donor, while Fatima will receive the kidney. So Fatima, she is the recipient. So the donor is Muhammad because it's the one that donates, and the recipient is Fatima because Fatima will receive this kidney. Okay? So this is for the donor and recipient. Let's move now to the definition of some types of grafts. Let's start first with the first type of graft. It's called autograft. Autograft is the graft that is transferred from one body to the same one side of the body to another side in the same body. For example, from my hand to my thigh, it's within the same body, so it's called autograft. While a non-example, from my hand to my friend's hand, this is not example. So autograft, this is a graft that is transferred from one side to another in the same body. So it's the graft that is transferred from one side to another in the same body. It's called autograft. Example, from my hand to my thigh, from my thigh to my face, let's say, it's called autograft. While non-example, from my hand to my friend's hand, this is a non-example. So this is about autograft. Let's move now to isograft. Iso, identical. So it's the graft that's done between two identical twins. So two identical twins, and not fraternal twins, identical twins. So the graft is transferred from one person to the identical twin of this person. So Samir's kidney transferred to his identical twin. So this is called an isograft. While non-example, Samir's kidney transferred to his sister twin. This is a non-example. So isograft is the graft that is done between two identical twins. Okay? Let's move now to the third type of graft. The graft is called allograft. Allograft, this is the graft that's done between person to another within the same species. For example, from Maya to Rilla, from a cat to another cat, from a dog to another dog. So this is called an allograft. The graft is done between different persons that belong to the same species. Is called allograft. Example about this, from Maya to Rilla, this is an example. And non-example, from a cat to a dog. Why this one is not example? Because from a cat to a dog, they are different species. From a cat to another cat, this will be an example. From a cat to a dog, this is a non-example because it's transferred from one person to another. This in the same species. Here we have different species, cat, dogs. And the last type of graft is, let's move now to the graft which is called xenograft. So xenograft is the graft that's transferred from one animal to another from different species. For example, from a cat to a dog, different species. While from a Maya to Rilla, they are same species. So this one is called allograft. While from a cat to a dog, from a cat to a human, it's called the xenograft. Here we move to Chapter 1, Document 3, Bob Meiosis. Biology is known to be the only science where multiplication and division, they have the same meaning. Mitosis is the process by which the somatic, the diploid, two end cells reproduce, and they produce new daughter cells. They are identical to the mother cell, having the same genetic information. So why the body cells need to reproduce? First, to increase the cellular number and allow the growth, like the case of the zygote, development into an embryo, and to renew the body tissues and replace the old, the dead, or injured cells. Mitosis is a set of events. It may be divided into interphase. We have G1, S, G2. G1 is gap 1, S it's the synthesis where DNA duplication took place, and G2, gap 2, where it prepares itself for mitosis. So mitosis is composed of MAT, P, M, A, T, prophase, metaphase, anaphase, and telophase. So this is the phases G1, S, DNA synthesis, and G2, and then we have mitosis. So DNA occurs and DNA replication occurs during the S phase. G2, it's as I said, is preparation for the mitosis, where we have two daughter cells identical to the mother. Here the chromatin during the end of interphase. We have chromatin form, and we have centrosomes that later will change into the asters. So during prophase, we have nuclear membrane disintegrates, mitotic spindle fibers are formed, centrosomes they change into asters and they move to the opposite poles of the cell, and the chromatin condenses so that each individual chromosome is made up of two sister chromatids. Here, metaphase is known as equatorial plates. The cells line at the middle of the cell, forming what's known by an equatorial plate. During anaphase, the spindle fibers they pull the sister chromatids apart, where we have two groups, each is made up of one chromatid each. So the spindle fibers they pull the sister chromatids of each chromosome apart. Each chromatid will move to the opposite pole of the cell. This is special about anaphase, is called polar ascension. At the end will be two groups, each are made up of one chromatid. Telophase and cytokinesis, where we have here the cleavage furrow, what's known by the cleavage furrow, when the cell cleaves into two identical cells. So the chromosomes they form two identical groups at the opposite poles of the cell. These will condense back into chromatin form. Nuclear membranes reform around each set. The spindle fibers disintegrate and the asters return to the centrosome state. Cell membrane constrict, forming cleavage furrow to divide the mother cell into two identical daughters. You have to know that the cleavage of the cell is known as cytokinesis, and the daughter cells obtained after each cycle they are identical to each other and they are identical to the mother. Now we move for meiosis. Meiosis is the process by which the mother cell of gametes diploid 2n will produce haploid gametes 1n. So before we start the meiosis, it should occur the interphase. Interphase is the phase that precedes the meiosis and it occurs only once. So we have the first one is called Meiosis I, it's called reductional division, and we will see why it's called reductional. And Meiosis II is called equational division, and also we will see why it's called equational. So Meiosis I and Meiosis II are preceded by interphase, and there is no interphase in between. So Prophase I. We have the formation of tetrads, where the chromosomes they pair up, they synapse. So all the processes are similar to mitosis. Nuclear membrane disintegrates, spindle fibers start to form, centrosomes change into asters, chromatin condense so that individual chromosomes are easily distinguished, and homologous chromosomes they pair up, they synapse, forming what's known by tetrads. This increases the chances of the crossing over between the non-sister chromatids. Metaphase I, the chromosomes they line at the middle of the cell. But here we don't have chromosome. What do we have? We have tetrads. So tetrads they at the middle. So tetrads they line up at the equatorial plate of the spindle at the equator of the cell. Anaphase 1. What will happen? The spindle fiber will pull the chromosomes the homologous chromosomes of each tetrad apart. Each chromosome will move to the opposite pole of the cell, forming what's known by polar ascension. The late will be two groups of chromosomes, each made up of two chromatids at the opposite poles of the cell. Finally, telophase will have cell membrane will constrict, forming what's known by cleavage forward. This is the key word during telophase. Two daughter cells are obtained. Each carries one copy of each chromosome made up of two chromatids. Each the obtained daughter cells, they are haploid, they are 1N, and the number of chromosome is reduced. That's why it's called reductional division. That's why meiosis I is called reductional. Daughter cells, they are different from each other and they're different from the cell. So have to know that no interphase will occur between meiosis I and meiosis II, and you have to know that chromosomes form two groups at the opposite poles of the cell. Then the spindle fiber will disintegrate and the asterns will return back to the centrosome states and nuclear membrane reform. Prophase II, what happens? Here we move to meiosis II. We have results. So the mitotic spindle fibers start to form, centrosomes change into asters, nuclear membrane disintegrate. Metaphase II, chromosome, they line up at the equatorial plate of the cell, at the equator of the cell. If you realize here, the same steps, meiosis II is very similar to meiosis I is meiosis II is very similar to the mitosis. So meiosis II, Anaphase II, what will happen? Spindle fibers will pull each chromatid apart, two groups of chromosomes, each made up of one chromatid. This is called by polar ascension. At the end, telophase and cytokinesis. We have telophase and cytokinesis. So we have the telophase II. We have four daughter cells are formed, and the chromosomes will decondense back to the chromatin form. Nuclear membrane starts to form around each set of chromosomes. This table is showing the comparison between mitosis and meiosis, where the mitosis, its aim is the growth and renewal and replacement of the cells or dead cells or the old cells, while meiosis is for the production of the haploid gametes 1N. Mitosis, one division producing two daughter cells, whereas meiosis, two successive divisions producing four gametes. Mother cell 2N and daughter cells 2N, the same number of chromosomes per cell, whereas number of chromosomes is reduced into half, going from the mother cell to the produced gametes. Mitosis, it occurs only to somatic cells in all the body organs, while meiosis, it occurs to the mother cell of gametes and the gonads. It's made up of one division preceded by interphase, which is the mitosis, while meiosis is made up of two divisions preceded by interphase. No pairing of homologous chromosomes in mitosis, where we have pairing of homologous chromosome leading to the formation of tetrads in meiosis. We have daughter cells obtain 2N, one chromatid per chromosome, while here, after meiosis I, two chromatids, 1N, two chromatids per chromosome. After meiosis II, 1N, one chromatid per chromosome. And you have to know that meiosis II is very similar in its events to mitosis.